just, for better visualization, what restriction enzyme NDE1 will do is an enzyme that will go in and look at any DNA, whether plasmid or regular human DNA. It'll look for this particular pattern. When it finds this pattern of 6 sequences, 6 bases, it's going to cut here and here to create what is called sticky ends. This part. See, you create these stepladder type shapes, these sticky ends here. We call these sticky ends because these non-overlapping regions right here are like puzzle pieces. They will bind with any other puzzle piece that has the exact same, that has the corresponding base pair pattern. So this overlapping puzzle piece, AT-AT, is going to look for any other puzzle piece that will be TA-TA because they will match together. And that can only happen when a restriction enzyme makes this type of cut, which is a staggered cut or a cut to make a sticky end. All right? So these ends, this restriction enzyme as an example, will go into the plasmid and make this type of, these 2 puzzle pieces here. And they will go into any gene that you want and make the same 2 puzzle piece ends. And then you can fit them together. So depending on what restriction enzyme you have, it will look for different patterns. And you want to use the restriction enzyme that will create the correct puzzle piece ends around your gene and the plasmid that you are targeting. Okay, so that's the main idea here, is that this restriction enzyme opens up the plasmid to create puzzle piece ends, creates those same puzzle piece ends in the gene that you want to incorporate, and then you fuse them together. And then you trick the bacteria into synthesizing the gene that you've incorporated into it. So, a very common question that is asked on the exam is something that looks like this. They will give you this diagram. It may be modified compared to the version that you're looking at right now. I'm going to go over some possible modifications in a second. And they will ask, give a possible strategy to clone this gene into that plasmid. And you'll notice the answer choices are like, use 1 restriction enzyme, use 2. Sometimes using 3 is okay. There's many different combinations this can be done in. Your job is to figure out which ones will actually work, which ones won't work, or which one works the best, or if that is asked. So, I went over some possibilities last time. I'm going to go over the more general possibilities that you can look at with this question. So, okay. So, I have a simplified version of a plasmid over here, drawn on the right, and I have sites A, B, C, D, and E, representing site for fake restriction enzyme A, restriction enzyme B, C, D, and E. This gene down here, this part of the plasmid, is called your antibiotic resistance gene. Every plasmid that you are going to use to clone a gene into must have some type of antibiotic resistance gene. The reason why is that when you incorporate a gene into a plasmid, how do you know if the bacteria even bothered to incorporate that plasmid? You will know that it incorporated the plasmid if it also gained the resistance to a particular antibiotic. Okay? Now this isn't as dangerous as it sounds. Usually the antibiotic resistance is some weak antibiotic that we just use to filter out the correct bacteria from the incorrect bacteria. But this is very critical here. You must have an antibiotic resistance gene so that you know which bacteria in your petri dish has successfully incorporated the plasmid. So we have these sites here representing restriction enzymes A, B, C, D, and E. And we have this gene here, this human gene here. Let's pretend it's the insulin gene. And they have similarly labeled spots for these restriction enzymes, except they're in different orders compared to in the plasmid here. The 5' end of the gene is labeled, and the 3' end of the gene is labeled, which will not always happen. If they do not label which end of the gene is 5' or 3', make sure you remember that the left side is 5' and the right side is 3'. And on the plasmid, This is the orientation that they order the plasmid in, is that the A restriction enzyme is more on the 5' side of the plasmid, and the 3' end is down here by the E site. I know the plasmid is circular, therefore how can we say one side is 5', one side is 3'? They're saying relative, these restriction enzyme sites relative to each other. The A site is 5' compared to the E site, and the E site is 3' compared to the A site, so it's all relative to each other. Okay, now that we broke that down, let's see what combinations of restriction enzymes may or may not work in this situation here. Okay, 1. If I just use restriction enzyme A, could I clone this gene into the plasmid? Well, in order to figure this out, what you need to check for is that restriction enzyme A will cut out the gene, meaning it will need to cut on the left side of the gene and on the right side of the gene, and you must make sure that A cuts somewhere in the plasmid. Okay? Well, A does cut here, so we'll open up the plasmid. That's step 1 done. And 2, does A cut both the left and the right side of the gene? Yes. A is right there, and A is right there. So A will successfully clone the gene into the plasmid. Just A by itself will successfully clone the gene into the plasmid because it prepares the 2 puzzle piece ends of the gene and opens up the plasmid very nicely. What about if we use 2 different restriction enzymes, A and B? Well, it's the same rules. You need to make sure A and B are cutting the gene and also cutting the plasmid. Well, A shows up right here, so that's good. And B shows up right here, so that's good. So A takes care of the left side of the gene, and B takes care of the right side of the gene. Notice there's also an A site here, but we don't care about that because B is right here. All right, and I'll explain us a trick in a second. But yeah, A is on the left, B is on the right, so far so good. Are those same sites in the plasmid? Yep. A is right there, and B is right there. It is very important that you pay attention to the direction that these are in. Whenever you use 2 restriction enzymes, you need to make sure they're in the same direction, both on the gene and in the plasmid. Well, A is on the 5' end of the gene. You check the plasmid. A is also on the 5' end of the plasmid. Good. B is on the 3' end of the gene. B is on the 3' end of the plasmid. Good. That means it will always insert the gene in the correct direction into the plasmid. That is very important because sometimes, I believe I have an example in a second, it will not insert in the correct direction. So A is good because A cuts on both sides and A is in the plasmid. Using A and B is okay because A is on the left here, B is on the right here, and they are in that same direction in the plasmid. What about A and C? Well, if we're using our rules so far, well, A is on the left here and C is on the right. That's good. And then on the plasmid, A is on the left here and C is on the right. That sounds good also. This trick I did not go over last time, so I'm going to tell you right now, this will not work, and here's why. Well, here's A, here's C, and there's A and C there. If you use restriction enzymes A and C, what you will create is the following. You'll create a fragment that starts as A, then you have your gene, then you have a fragment that ends in A, plus you have this little tiny piece that starts as A and ends as C. How did that happen? Well, yes, A is on the left and C is on the right, but A right here is a little bit closer to the gene. Okay, I'm going to contrast this with number 2 again in a second so you see the difference between number 2 and number 3. But in number 3, when we use restriction enzyme C here, because it's further away from the gene, the fragment you will actually make is a fragment of the gene with an A end on the left and an A end on the right, because this A end right here is a little closer to the gene. You will also create this useless fragment here representing the space between A and C on the right. So, if you do that here, and you opened up A and C into the plasmid, because you're using 2 restriction enzymes, you'll be opening up both A and C in the plasmid. You cannot insert this gene into this plasmid because there's too many holes in the plasmid, and this gene right here has 2 A ends instead of 1 A and 1 C end. In fact, probably what's going to happen is that this thing, which is useless, is actually going to be inserted into the plasmid because it has a corresponding A side and a C side. So number 3 cannot work, and that is critically because this C side here is too far away from the gene compared to the A side. Let me contrast that with number 2 so you see the difference between number 3 and number 2. If we do number 2 again, where we use the A and the B restriction enzymes, what fragments will we make? Well, we will have a gene with an A fragment on the left, that's good, and an A puzzle piece on the left, and we will have a B puzzle piece on the right, because look how close it is to the gene compared to the other A site. We will create that. You will technically also create a useless piece of fragment that doesn't matter, a B and an A thing, but that doesn't matter because that doesn't have the gene that we care about. So you will still, in number 2, create the correct gene fragment. That would, because it has an A on the left and a B on the right, will successfully insert itself into the plasmid. Okay, again, contrasting that with number 3. With number 3, you really need to pay attention to where all these sites are relative to each other. Because C is so far away, in fact, it's further away than this A site here, you will create these fragments instead, because they're not just going to ignore, the restriction enzyme A is not just going to ignore that site. It's going to cut that site as well to create this messed up situation. This gene will not be cloned into the plasmid because it's 2 A sites, when according to number 3 here, you created an A and a C site. All right, so be very careful. I did not go over this potential trick last time. You must not only pay attention to, oh, one is on the left and one is on the right, you must also pay attention to where they are relative to each other, okay? Moving on to number 4, and I will go back and explain number 3 again if people want me to, but for number 4 for a second, it's a little bit easier. Could I use restriction enzyme A and D? No. Why? Well, let's see. A is here. That sounds good. But D is here, and there's A also. So it sounds like D is on the left side of the gene, and A is on the right side of the gene. What is it on the plasmid? It's the other way around. On the plasmid, A is supposed to be on the left, and D is supposed to be on the right. So on the gene, it's backwards, so you cannot use this. Okay? So A we can use, number 1 I should say, number 1 we can use, number 2 we can use, number 3 we can't, and again number 3 we can't because of this weird situation over here. Number 4 we can't because everything is backwards, D is on the wrong side compared to A in the gene. And lastly, just to finish this off, could we use just restriction enzyme B by itself? No, because what does restriction enzyme B do? Restriction enzyme B only cuts one side of the gene. We can't cut the other side of the gene. Yes?
So when you're saying like A and B, that's not like order? Like you can do A, then B, or B, and then...
Yeah, when you put it in the experiment, you don't really do it in any order. You put them both in, so there's no way to tell which one happens first or second. If that's what you're asking. There's no real way. You put both restriction enzyme A and B in there to prepare the gene. Yes?
For number...
example where it goes backwards?
Backwards would be number 4 here. This one would be a backwards one.
Number 1.
Number 1 will go backwards half the time. So yeah, I forgot to mention that. Number 1, because you're using one restriction enzyme, you create a front and back end indistinguishable from each other because you use the exact same restriction enzyme at the front and at the back end. Therefore, it will work. If the question is asking, does it work? Yes, technically this works, but it will only work 50% of the time. Okay? Whenever you use 2 restriction enzymes, it will work 100% of the time because you've created a front end and a back end that is distinguishable from each other. You have a question?
For number 3, if the 3', like the C and the A were flipped, could you still go with it?
Yes, that's good. Is that if C was a little bit closer compared to A, you could use it. Because then you would ignore the outside A. The outside A doesn't matter. It's like what happened for number 2 when we used A and B. Well, when we used A and B, we could ignore this one because it's further away from the gene and we don't care about that. So same thing is if C and A were flipped around, we could use that.
How far is too far?
What I mean by too far is relative to each other.
Okay.
Yeah, what I mean by that is relative to each other. Like if C was closer to the gene compared to A, you're good. That means we can ignore the outside A. I guess a short rule of thumb is go with whichever 2 spots are as close to the gene as possible. When you're given like this situation with A and C, and you notice there's 2 A's and 1 C site, pick the 2 spots that are as close to the gene as possible, and then see if it makes sense. Originally, when A was here, when we did A and C, and A was right there, well, here's A, and there's an A and a C site here. Realistically, that means only the 2 A sites would be used because they're closer to the gene. Contrast that with an A with number 2 here, where you're using A and B. Well, here's an A, but then here's a B and an A on the right side. You need to use the 2 closest to the gene, A here and B there. That was good. Okay. Yes.
Would there ever be a time you can go from like 3' end to 5' end and then have like C and A and have the gene incorporated?
Sorry, I'm not really sure I understand. You're saying that if the C was over here or
No,
something?
like leave it the way it is, and then have it go from 3' to 5', and then you can incorporate the gene. If you go C to A, would that be possible?
That would be putting it in backwards. You're saying the exact same orientation, but you use C and A.
Or like, I don't know, somehow to, instead of going like A to C and having that work, or like not, but like if the other A was gone, going C to A.
So if this A was gone,
Well, okay, so like in
Sure.
the sense that you have it right now, the A isn't gone, but then if the A was gone, oh, no,
what would happen? You're saying if this thing was gone, what would happen? Yeah, if that A wasn't there, it would work. Yeah. Yes.
A is both on the 5' and 3'.
Yeah.
Why not the same thing to the plasmid that we see in the 5'?
It's all relative. So you're talking about if we just use A by itself?
Yeah.
Yeah. So if you just use A by itself, you've created a left and a right side here. For the plasmid, when you only cut this one A site, because there's only one A site here, you will already create the sticky ends that you're looking for. You don't need to cut twice in the plasmid because it's circular. You don't have to worry about that. Right, it does not have to do, if you're just using A, you don't have to cut A twice. Cutting it once is fine. Only when you use 2 restriction enzymes, you need to cut, one of each on the plasmid so that they match together. Okay. All right. Anything else? I know it's a bit messy.
Will there ever be a repeat of enzymes on the plasmid?
Yes, actually, sometimes you will get a restriction enzyme that will cut multiple times on the plasmid. I haven't seen that example show up on the exam, but if so, let's say what happens if there's an E site here, an E site here, an E site there, and let's even say that E is correct. E will show up here and here. Could we use restriction enzyme E? The answer is no. Why? Well, you can prepare E here and E here, but man, the plasmid's going to be ruined when you use restriction enzyme E on the plasmid. It's going to open up holes everywhere. You don't want to do that. We want to specifically use a restriction enzyme that cuts either just one spot, or when it uses 2 restriction enzymes, they better be close together. I've drawn it here, and it kind of looks like it's farther apart. Let me go back to the picture so you see what I mean. But realistically, look at how close the restriction enzyme cuts are actually to each other. So there's regions in plasmids, forget what they call it, but they're kind of cloning regions. They are really special regions that for whatever reason have lots of variety in terms of restriction enzyme cut sites, and they're all crammed close to each other in the plasmid. So in our example on the drawing here, we wouldn't use restriction enzyme E because it cuts it too many places. It cuts it at too many places, and you basically destroy the plasmid if you do that. But using just restriction enzyme A by itself or B by itself, no, B by itself would work. But just using A by itself would be fine. Okay. Because it only cuts once on the plasmid. Other questions? I think that's every possible combination that you'll get out of this style of question. So feel free to watch the video again when it's ready. Sure. This better not have stopped. Oh my god. When did it stop? Let's see. I guess I'll record it at home on my own. Alright.
If you want, I can airdrop mine on
Oh, you have it? Alright, we can do that later. Alright, let's turn this back on. Release. covered in lecture, so I'm going to briefly talk about chapter 11. Alright, so chapter 11 is a very short chapter. There's really not a lot that you have to worry about, so I'll just cover the important parts. One, and I still have to post the chapter 11 worksheet, but it's a really short chapter, so I'll fix that tonight. Chapter 11. Very first thing they talk about is the different types of DNA that you have, A, B, and Z DNA. Again, I'm not going to go over it too much. B DNA is the really common one. Z DNA is the less common one, and A DNA you never see in nature. There's some numbers associated with it, which you can look at, I believe, at the end of chapter 10b, such as base pairs per turn. I'll ask the most important one. How many base pairs per turn are you going to find in B DNA? 10. 10 base pairs per turn, so make sure you memorize that. That is because, eventually when I get down to it, we're going to talk about supercoiling. So, before I get down to it, some things that you can do to DNA. One, you can melt DNA. You can denature DNA. How do you do that? You just increase the temperature enough, and every strand of DNA will eventually separate into two individual strands of DNA. Every duplex double helix DNA will eventually break apart. They will break apart at different temperatures depending on how many G's and C's they have, right? You remember how many hydrogen bonds G's and C's have? How many? 3 hydrogen bonds for G's and C's, meaning any particular DNA sequence that you have, if it has a lot of G's and C's in it, will have very strong hydrogen bonds. Therefore, in this graph, you can see here, some organism that I don't know how to pronounce has 66% G's and C's. Notice that it requires a melting temperature somewhere of 95 degrees Celsius before it can actually separate. Compare that to pneumococcus, who's only 38% G's and C's. It is a lot easier to separate it at lower temperatures. How do I know that this means it's separating? Well, remember that DNA is composed of nitrogenous bases, which are aromatic. And you know by now that aromatic compounds absorb what? UV light. What this graph is telling you is that when DNA separates into two separate strands, all of a sudden its ability to absorb UV light increases tremendously. That's because those nitrogenous bases that were hidden on the inside of double-stranded DNA are now open to the world when DNA unwinds from itself. So we can tell when DNA is melting when all of a sudden it absorbs a ton of UV light at a particular temperature. This whole mechanism here, this whole experiment is called a hyperthermic shift, and that's how you can identify at what temperature does a DNA sample open up. Two other things that you can do to DNA. One, well, once you melt it apart, you can actually let it recombine on itself. If it does this very quickly, if the rate of renaturation, recombining is very fast, it is very simple DNA. It's a very simple line of bases. Like you will get long stretches of A's and long stretches of T's and long stretches of G's and C's. That type of pattern will recombine very easily. Contrast that with a very complicated DNA sequence, it'll take a lot longer. Hybridization, very simply, is you take two DNA strands from different organisms and you see how well they base pair together. If they base pair very well together, they are more homologous to each other. They are more similar to each other in terms of their genomes. So you can create phylogenetic trees based off of DNA hybridization. All right, now we get to supercoiling. So, what supercoiling is when you take DNA, and for whatever biological reason, sometimes it's twisted too much and sometimes it's not twisted enough. When it is not the right number of twists, when DNA is not in the right number of twists, it is in a supercoiled state. It is either positively supercoiled or negatively supercoiled. How can you tell if something is positively or negatively supercoiled? You can solve this formula. Linking number equals twists plus ride, or rid, or whatever it's pronounced. So make sure you memorize this formula. What this formula is the main thing is linking number. When you look at the linking number, you can figure out if something is positively supercoiled, negatively supercoiled, or in a state that is called relaxed. When a piece of DNA is in a relaxed state, it is at the perfect amount of twists. How do I know what the perfect amount of twists is? Well, here's an example down here. They are given a DNA sample that is 400 base pairs long. They want you to figure out what are the numbers associated with this when it's in a relaxed state. When something is in a relaxed state, W will always equal 0. Okay, so step 1. When something is in a relaxed state, W equals 0. Right or rid equals 0. After that, you can calculate the linking number in the twists, and here's how. You can always calculate the number of twists that there are supposed to be. And the way you do that is you take the total number of base pairs that DNA has, and you divide it by 10. Why do I divide it by 10? Well, we said earlier that B DNA, which is the most common type of DNA that you have, B DNA is 10 base pairs per turn. So because you know how many total base pairs there are, and you know how many base pairs per turn there are supposed to be, you can do some division to figure out how many twists that there are supposed to be in a sample of DNA. 400 divided by 10 base pairs per turn gives us 40 turns. So we can, no matter what, calculate the number of turns there are supposed to be. We are told that W equals 0, that means it's relaxed, so we can plug these two numbers in here and we calculate the linking number. Meaning for this particular question, when the linking number equals 40, it is relaxed. If you are given a second piece of information saying, okay, now that you've finished calculating what the linking number is supposed to be, you find out when you actually measure it that the linking number is 36. What type of supercoiling is this DNA sample in? Negative supercoiling. If you actually measure, you can calculate what linking number is supposed to be. But then when you actually measure the linking number and you find out it's 36, you know that there must be some negative supercoiling because the linking number is less than expected. That will be given to you in the problem. Like they will tell you the linking number actually is 36. What's going on? Negative supercoiling. It's less than it's supposed to be. They might say the linking number now is 45. That is positive supercoiling because it's higher than it's supposed to be when you compare it to the relaxed state. It's as simple as that. All right. You can sometimes have drawings with these, but don't worry about the drawings. We'll just straight up give you the numbers. So if the drawings are a little bit confusing, don't worry about it. So I'm going to skip that. All right. Next thing in chapter 11 they wanted to address is a special type of structure that DNA can sometimes form, and that is called a cruciform. A cruciform is a special DNA structure that will happen that will create this like plus shape here. Basically, what ended up happening is a bit of a mistake. One strand of DNA up here has decided to temporarily bind to itself rather than the opposing strand of DNA. Regular DNA is when one strand binds to the other, but a cruciform is when one strand of DNA binds to itself rather than the other strand, and it will do so on both sides right here. It creates this plus shape, this cross shape. And on the exam, a very common question that is usually asked is, they'll give you 4 different double-stranded DNA sequences, and they'll ask which one will form a cruciform. So how do you know if a sequence is going to form a cruciform? What you need to look for is what is called a palindromic sequence. What a palindromic sequence is you read a particular sequence from left to right here, then you go and check the very opposite side, and you read it the opposite direction, from right to left. If it is the same sequence in both of those situations, that is a palindromic sequence. So let's check here. G-A-A-C-G-T-C-C. When I read it from left to right, I go all the way down here now. G-A-A-C-G-T-C-C. That is the same exact sequence, but they're going in opposite directions. That is a palindrome, okay? So you kind of have to look crisscross on this picture here. Notice that you don't have to have the entire thing follow the palindromic pattern. Notice this middle area will not be part of the palindromic sequence. That's fine. You just need at least 2 opposing parts that are following a palindromic sequence. As it won't be color labeled on the exam, so you'll have to go and look at the different patterns and see which one will form a palindrome. If you find one, it will form this nice shape right here. A variation of this type of question, when you see it on the exam, will be which one will form the strongest cruciform or which one will form the weakest cruciform. Step 1 is to figure out which sequence even forms a cruciform in the first place. And then step 2 is the strongest cruciform is the one with a bunch of G's and C's in it, right? Because there's more hydrogen bonds. The weakest cruciform is the one with a bunch of A's and T's in it. That is because there's less hydrogen bonds in there. Okay, so The next bit of information only applies to eukaryotes. Only applies to eukaryotes. Prokaryotes do not have this thing. So in eukaryotes, DNA genomes are usually a lot longer. There's way more base pairs in eukaryotes, in a eukaryotic genome. So if they want to store that DNA in eukaryotes into a small compact space that is the nucleus, what eukaryotes have developed are these things called nucleosomes. So what are nucleosomes? So nucleosomes are these structures here where DNA is wrapping around a set of proteins called histones. Okay, so these yellow things on here represent a spherical combination of protein subunits called histones. These histones will have DNA wrapped around them in order to store DNA in a nice compact area, because you're wrapping things around a small area here. So what a nucleosome is DNA wrapped around a histone protein. That's what the definition of nucleosome is. It's DNA wrapped around a histone protein. Okay? So that's the first level of complexity with DNA structure, chromosome structure in eukaryotes. The next bit of complexity is actually illustrated in this diagram here. They use the phrase epigenetic marks or epigenetic modifications. So, in eukaryotes, after talking about DNA being wrapped around histones, and that's how you create nucleosome structures, there's a couple extra things that are usually added. Epigenetic marks such as methylation, and it's not, oh yeah, it is listed here, or acetylation, and I guess phosphorylation is included also. I'm only familiar with methylation and acetylation, are slight functional group modifications to nucleosomes that will change a few things. If a particular nucleosome or region of DNA has a lot of methyl groups on it, it is very hard to unwind that DNA from histones. Why do we even need to unwind DNA from histones? Isn't it great that they're stored on nucleosomes, on histone proteins, right? Well, if you want to actually transcribe a gene, if you want to transcribe a gene and then eventually translate it into a protein, you need wide open DNA that isn't wrapped around anything. Okay? So nucleosomes are more of a storage thing. A nucleosome's job, a histone's job is to store DNA in a compact area. However, if you want to transcribe that DNA into something useful, you need to slowly unwind the DNA away from the histone, and then you can transcribe it with various enzymes. So, depending on what that nucleosome is like, sometimes it's easy to unwind the DNA from histones, from nucleosomes, and sometimes it is difficult. The times are when it is difficult to unwind DNA from these histone proteins is when there's a lot of methyl groups, okay? So whenever you hear, at this point on, in this class or other classes, when they talk about DNA methylation, they're talking about genes that are not easily transcribed because they're really bound to the histone protein. It's very hard to unwind the DNA from those histones. The opposite of DNA methylation is acetylation. If they talk about a region of DNA that is very well acetylated, it's a lot easier to unwind DNA from these histones. If you dive right into that in terms of organic chemistry or polarity type chemistry stuff, well, a methyl group, is that a very polar or non-polar thing? It's non-polar. Methyl is CH3, it's a non-polar thing. So that actually makes it harder for anything to interact with the histone to try to unwind the DNA from it. Acetyl group is in part a carbonyl group, which is a carbon double bonded to an oxygen. That is highly polar. It is easier for things to interact with an acetyl group, therefore it's easier to unwind DNA from it. So again, this pattern of methyls and acetyls and regions where there isn't a lot of methyl groups, hypomethylation, will affect how genes are transcribed in a eukaryote. This figure is really annoying. But basically what it is, I see we're running out of time, so I'll talk about this more in detail next time. This figure is just telling you that there are enzymes that exist that mess with nucleosomes, that shift nucleosomes back and forth so that sometimes you can transcribe a certain gene very actively and sometimes you can't. So I'll talk about this because this is sometimes an exam question also.